Alright guys, let's start. Again, so for those of you who haven't been here on Tuesday, so I'm Tobias Hamp. I gave the lecture on Tuesday and will be giving this lecture today because Burkhard is currently traveling. But he will be back next week. My topic is the homology-based prediction of protein function. Just a second. Nope. Pointer works, switching doesn't. All right. Topic is the homology-based prediction of protein function. A short recap of the last lecture. So I explained a very simple way to predict function from sequence by simply side-blasting your sequence through a database of annotated sequences and taking the best hit that you find and transferring all the functions of the best hit to your query. You can use this for EC numbers, for localization, cell cycle control, and many other functions. You see that this sort of procedure works very well for in case you find proteins that are very similar to your query protein. So almost no mistakes until a region of around 80% sequence identity. For more distantly related proteins, but you will just then, however, need more advanced methods, for example, based on machine learning and motifs that you will learn in the upcoming lectures. I explained that about 3 years ago we had an exercise in the context of this lecture, where we ask students to implement methods that uses a so-called k-nearest neighbor method to predict protein functions. So we are now not only looking at the one best hit that we find, but also on the k best hits, and also introduced weightings of the k hits. And what we would ideally want in the end are 3 types of annotations for each protein that we predict, namely one time what exactly it does on the chemical level, in the context of which process these functions are happening, and where in the cell and in which complex it all takes place. And I introduced very extensively the gene ontology. So gene ontology again defines 3 separate graphs for each protein. On the I have to say they're rooted graphs, and in the root is the ontology itself. And as you go down the hierarchy, the terms become more and more specific until you reach the definite functions at the very leaves. I explained 3 different methods for the weighted k-nearest neighbor scheme, and this is one of them. So this method first of all calculates the so-called quality score for the overall hits that you have found with your side-blast query. It calculates a go-term specific score, and it finally multiplies the 2 scores in order to be in a range from 0 to 1, and for the scores to be comparable not only within the same query protein, but even across query proteins. I also explained an evaluation measure, which is used to really find out how well you predicted in the end. For example, you take one cutoff, which reduces your prediction to two terms in this case. You compare how accurate your predictions then are. So in this case, two of them are correct. You you look up how much is covered from all the correct terms in your prediction. In this case, it was 10%. And both values are referred to as precision and recall. And if you repeat this for one protein and many thresholds, you get a recall precision curve for this one protein. You repeat this again for many proteins and you get multiple curves. You average over all the curves to get one curve for your entire method. And if you want to boil down your entire method to one point, you calculate the so-called maximum F1 score. And as a warm-up for today's lecture, I want you to find flaws in this measure. So of course this is not the only way you can measure function. And if you were a user of any function prediction method and you would find such a curve or such an F1 score, what would you think? So what would come to mind that is not ideal with this measure? Let's say 5 minutes.
Question? So
yeah.
we only get to see the F1 score?
No,
the curve.
The curve and the score.
Okay.
Wenn man jetzt bei dem einen so gut ist und dem anderen richtig schlecht, dann ist im Durchschnitt trotzdem nicht okay. Aber trotzdem ist das sehr gut. Aber sieht man das an der Kurve?
Ja, in der Kurve kannst du es nicht sehen.
Ja, an den einzelnen aber schon.
Wenn du alle hast.
Ja.
Aber du siehst ja nur die schwarze.
Wer sagt das, dass nur die schwarze ist?
Weißt du, wenn du ein MCC machst, zum Beispiel, dann siehst du auch keinen Standardwert.
Nee.
Ja, aber dann hättest du praktisch den Durchschnitt, dann kannst du den Durchschnitt besser finden.
Ja, das ist ja auch eine Durchschnittsstation.
Ja, das schwarze ist der Durchschnitt. Ich glaube, es gibt bei dem F1 auch so allgemeine Nachteile, aber ich kann mich da nicht mehr erinnern. So allgemeine Nachteile gibt es so. Ja, wenn man jetzt einen sehr unausgeglichenen Datensatz hat, dann funktioniert das vielleicht nicht mehr so. Aber weiß ich jetzt nicht sicher. Da gibt es doch so Messungen, so Kennzahlen, die dann nicht so gut sind.
Ja, genau. Ich weiß es jetzt gar nicht mehr.
Genau, du hast ja keine Verhältnisse von True Positive zu True Negative zum Beispiel. Oder also das, was du MCC
Ja, das ist aber letztendlich Precision Recall.
Ja, schon, aber wie viel Precision?
Precision ist, wie viele der Recall ist, wie viele der positive du gefunden hast.
Und wie viele von deinen Vorhersagen?
Und Precision ist, wie viele von deinen Vorhersagen.
Ja, genau, du hast kein Verhältnis zwischen den beiden, oder? Also es kann sein, dass es...
Ja, das meine ich ja. Das könnte praktisch, der eine könnte richtig gut sein und der andere richtig schlecht. Und durch die Berechnung, ja gut, nee, eigentlich nicht, weil du nimmst mal, also das ist ja nicht nur.
Ja, also du findest nur ein Positiv und das ist auch true. Und dann, also dann kann es halt sein, dann sagt er ja 100% richtig.
Ja, aber das Recall.
Ja, aber du hast vielleicht total unausgeglichenen Datensatz. Kann man das auch nicht auslesen. Also so ein True Positive Wort einen Datensatz gibt. Und das ist schon anders mit dem.
Warum hat man denn nicht diese RC-Kurve? Das ist auch noch
Das hätte ich mich auch gefragt, ja. Aber kommt da nicht nochmal Recall Precision drauf?
Bei RC.
Recall Operator.
Mhm.
Also da hast du auf der einen Seite Recall Operator.
Da hat man
Das Specificity ist nicht so viel.
Aber
Ja,
Sensitivity
eine-
ist Precision, glaube ich.
Ja, aber einer geht ja nicht und du hast ja keine True Negative drin. Also Sachen, die du nicht vorhergesagt hast,
Ach so.
die nicht kommen, kannst du nicht nehmen.
Stimmt.
Also kannst du diese RC-Kurve nicht machen.
Ach so, weil du Specificity also das.
All right. So, okay. So, final answers. I heard some good concepts already.
Well, then I guess one point was that with only the average line, you cannot see what the yes the error bars or how much better or worse the function will be. So it could be like
Yeah.
every curve is like around this average, or it could be that some are very good and some are very bad.
Yeah, this is true. Yes,
So
Yeah,
possibility.
that's certainly true. Yeah, but it's somehow a necessary evil because you always have to average over the proteins, and not every protein is predicted, so it's equally easy to predict.
Yeah, but we
So-
could like show the standard deviation and
Oh yeah,
I don't know,
yeah.
like
Yeah,
two
it's true.
more curves.
Yeah, it's
and
true. So yeah, you
could.
yeah. So there are two issues involved with that. So first of all, so not every protein is as easy as all the other ones, and also as long as you do not have a large amount of proteins, you have sort of a large area. A large error in the curves, especially in the lower recall regions. And yeah, but yeah, let me show you one other example. So assume this is the root of an ontology, and those are two predictions where, let's say, this is correct, this is correct, this is correct, this is not, and this is not. How would you weigh these two predictions? So which one is better? Yeah.
So obviously, predicting the right one is more difficult.
Yeah, exactly. So this could be a very generic term. For example, the protein is an enzyme, which is like around the 30% hit rate. Whereas this could be a very rare function with only 0.1% of all proteins. So predicting this correctly is much more difficult than this one. So you would want to really weigh these up. Right. And yeah, you would want to incorporate also generally the depth, the specificity, but also yeah, it's a question whether you actually want to rate these things in the middle, because they are sort of implied by the other function. And it is often the case that, at least in practice, this function, you never find this function without that function. So they're also also correlated. Yeah.
Just a question, because I wasn't here last time and I wasn't even aware there was a lecture. Precision and recall are defined as the number of nodes we find out of the number of nodes.
So So precisioning recall is always defined for one threshold. If you take the 0.8 threshold here, precision is the fraction of predictions that are correct. 2 in this case. So this is the one prediction that you get right. This prediction is wrong. So precision of 0.5 in this case. And recall is the fraction of all the annotations that you could possibly have gotten correct. So in this case, you get 1 of all the other ones, which are 11, so around 10% precision. 10% recall, sorry. And, yeah.
But when you look at this long prediction,
Yeah.
Isn't it weighted like indirectly because all the other nodes are also, they have to be correct, right? When the nodes at the bottom are correct,
Yeah.
then the nodes in between are correct as well.
That's true, yes.
Yeah, so getting those 2 is not like 2 true positives, but like 5 true
Yeah,
positives.
exactly.
And the other one is only 1 true positive.
Yes.
So it's kind of weighted anyway, right? The more you go, the more deeper you go with your predictions.
Yes, but so both proteins contribute equally to this average curve here.
Yeah.
Right. So this means this prediction is just as important as the one here.
Okay.
So you
Okay, now I
yeah. So there are many superficial predictions. Many targets are only annotated superficially. For example, protein binding is an enzyme, something like that. And you would really want to measure those ones here. And one last big issue is also that this measure works actually, even if you predict the entire ontology, so all the 50,000 terms for each target. Because all that is required is that you give different scores to all the terms. Yeah, maybe you think about that at home in order to really understand it. But really, this is a big issue, because obviously, if you would then put the method online and the user would run it, and he would get back the entire list of Go terms that there are, basically, so 50,000 predicted functions, and then has to decide for himself which functions he actually wants to use. This is sort of very user-friendly, user-unfriendly. And so these are open issues with this measure. I will then shortly introduce another measure that corrects for this, but not in great detail, because it is quite complicated. So it has become quite complicated. But so this is just to show you that even the evaluation of function is still under construction, essentially. All right. So after we had developed the methods in the context of the exercises, there was this CAFA meeting. CAFA meeting had targets, had us predict 50,000 targets, which some of them then became annotated 4 months later. This is the sequence identity distribution of these targets to the ones that are already annotated. This part here is actually a rather natural distribution. So you would get this sort of distribution for all the sequences that are not annotated here. This part on the right here is rather artificial. So this comes from the fact that even experimentalists like to investigate proteins that are already that are similar to the ones that have already been studied. We performed rather mediocre in the MFO ontology, but for example, for difficult BPO targets, we performed really well, which is quite a good result for the methods developed by students in a couple of months, which were competing basically against the rest of the world. To summarize kappa, so all the methods that were used were sequence-based. No, not all the methods that were used were sequence, but the top performing methods all use sequences in some way, and high throughput data like protein interactions or expression contributed at least to the overall accuracy. Now, so we were quite encouraged by these results, so we tried to further improve our methods. So from now on, I will refer to the baseline classifiers as student A to student C. As I said, all based on SciBlast. Several issues with the method as we used them for kappa. So kappa always only evaluated one method. We had to choose the method, which was method A in our case, and there was another method C, which actually performed better. So this was a rather bad choice. There were various free parameters among the methods that were not optimized or optimized in a suboptimal way. We did not have time, so the students submitted the methods rather shortly before the kappa submission, so we did not have time to summarize the predictions in a meta predictor. One of the methods actually had quite a severe bug, which really was quite detrimental to performance. And finally, a small but also important detail was that we did not have the original implementation of this measure that I just explained available, which made a small but decisive difference. And also the evidence codes that were used by the gene ontology for the targets were not known at time of submission. So after kappa, we said, so what would have happened if we had done all these things correct in the first run? For that, we basically simulated a kappa 0 experiment. This was basically everything that would have happened if kappa had been conducted 1 year earlier. So we used kappa 0 to optimize our methods, and then evaluated them again on the actual kappa targets. And to put it into a more formal background, so we used everything that was annotated before 2010 as our templates. Targets was everything annotated in 2010. Both that's together were our training environment, and the final predictions were then just like for kappa itself. So from now on, I will refer to the training set as smaller than 2010, test set as 2010, training set for kappa 2011, and targets again 2011. The three parameters that we optimized were the number of hits that we consider, the e-values, so you could not only look at the best k hits, but all hits that are more, for example, more likely than e-value of 10 to the power of -3, and also other algorithmic alternatives that the students had provided themselves, for example, using sequence identity instead of the positive identity. And among all these parameter combinations, so we tried out all these parameter combinations, and we simply picked the one that performed best among all. And the optimized methods then called student A prime to C prime. Now we had our optimized baseline predictions. Now we had to combine them in a meta predictor. So following the scheme of the nearest neighbor principles, we chose a meta predictor that was really the most simple that we could think of. In this case, a linear regression. So this means if you predict one protein with all the baseline classifiers, you collect all the terms that are predicted. If a method, if a Go term is not predicted by all methods, this prediction has a score of 0.0 in all the other predictors. This way you have 3 numbers for all the goal terms in this particular target, which allows you to train a meta predictor. In this case, so A prime, for example, means the probability as output by method A prime. And what you then have to optimize is X, Y, and C. And in the end, you get a probability P, which combines, as I said, all the baseline classifiers into one score. One problem with, so one important detail of this was that of course in machine learning so it is not ideal if you use the same method for optimizing the baseline classifiers and for training the meta predictor. So we split our test set, our optimization test set, into 2 parts, and used one part to optimize the baseline methods, and then the other part for training the linear regression. And in this way, you switch the roles of these 2 the test sets, repeat the entire procedure, and then average over your weights. So it's in sort of a cross-validation kind of scheme. And yeah, without further ado, we go to the results. So you don't have to look at the details of differences of all the methods. Just look at the best performing ones here. So these squares here, light squares, are from the best method, namely the meta predictor on the optimized baseline methods. This method performs really well for intermediate levels of recall in the BPO ontology, which is on the left here, and for all levels of recall in the MFO ontology. Only method C here. So this was actually submitted to CAFA but not evaluated. Also performed really well for low recall regions in the BPO ontology. And yeah, one criticism of not of the recall precision curves that we just discussed earlier, but of the maximum F1 measure. So the way with which you really compress the curve to one point is that it performs extremely well for intermediate regions. So it is virtually impossible to get a good maximum F1 score only if you're only good in rather low or rather high recall regions. So it really favors any method that performs well in this intermediate region. This is also why you see sort of a bias towards this region, because so as our criterion for choosing the best parameters, we actually used the maximum F1 score. All right, any questions so far? Okay. So then we also considered one improvement over this way of rating functions by only looking at the leaf terms here, so by ignoring everything that is in between, and this basically measures how well we find these specific functions that the proteins have. And so really by only considering the terms, so everything else stays the same, but we simply drop everything that is in between the leaves and the roots. And this is here shown on the right. And as you see on the scaling here, so methods are hardly finding anything here. So you almost never reach over a precision of 30% and never over a recall of 20%, which also indicates really how difficult this prediction problem actually is. And also an important thing is that it really depends on the measure also, so that the order which method you perceive as best depends on the measure. So the order of methods in the leaf measure that I just explained is different from the one in the threshold measure. Summarizing these optimizations, so had we had these meta predictors a lot earlier, we would have been about only 1 percentage point worse than the best method in kappa. So we would have really done very well. For the general problem of function prediction and assessing the predictions, no single measure is enough. And depending on the measure and on the ontology, methods perform differently. Of course, the problem is still not resolved even in the slightest way, and the general problems of homology inference persist. So problems that you should have heard last week, for example, like the domain problem, so that the parts that you align, that you find to be similar between two proteins, are not actually the ones decisive for your function, or that you can even change function by only exchanging one amino acid. And this also brings us directly to kappa 2. So this is now a big time warp. So kappa 2 was held 3 years after the first kappa meeting. Reasons for this was that the kappa 1 meeting was a big success. So they had many new methods which they wanted to publish. And for kappa 2 then, we tried to do a few things better, but everything that I say now still is under heavy construction, because so the evaluation was at the end of July of this year, and people are still really looking at the results and trying to find the best way to present them, and some of the issues with the results I will show in a second. First of all, so what was different? So the gene ontology was not involved itself in the first kappa experiment at all, but they got wind of kappa, so they learned about kappa shortly after and tried to contribute themselves. So they actually held a talk at the kappa meeting then. So just for us, they introduced the so-called annotation blacklist, which you can use for automatic predictions, and which tell you which type of predictions do absolutely not make sense. So, for example, like this one. So in this case, a protein is very similar to a protein that is already annotated, but the only difference is a residue that is decisive for the enzymatic function of the template. And of course, if this one residue is different, then even the highest sequence identity doesn't help you with function prediction. And also, if you have a target that comes from an organism that is not photosynthetic, for example, and you predict functions which are involved in photosynthesis, also doesn't make really sense. Similarly, so-called taxon restrictions, which are Go terms that should not even be used for entire taxa, like for example, the flower development should only really be used in plants and not in humans or anything. And finally, some terms were discarded for automatic function prediction, like terms which only describe a function that is carried out for a very short period in the cell cycle, or Go terms which are generally considered to be too general. And if you compare this to the rules that were already in place for the first kappa meeting, and compare this again to what we used in our preliminary evaluation of the methods, you will find like a difference. You go from 10 lines of code for the gene ontology to around 1,000 lines of code, and who would have thought that? Also, there's now a new discipline in kappa 2, which not only measures the prediction accuracy for targets that that did not have any annotation so far, but were already annotated and gained new annotations. We used a new evaluation measure that is based on information theory and semantic distances, which is quite complicated. So it would take a half an hour probably to explain it, even only in a coarse way. But it really accounts for all the issues that I just explained. So information theory is used to specify how much information content really is in one prediction. So if one Go term always co-occurs with the other Go term, then this should only account for half of the overall weight that the prediction has, for example, or if one protein is annotated with a lot of functions, this should be rated higher than a protein that is only, for example, annotated with enzymatic activity. And finally, we used again two evaluation modes in order to also consider methods that predict, for example, only proteins in human. So we evaluated on all common, on all the targets for which all the methods made predictions, and we did this in one mode, and in the other mode we penalized unpredicted targets with a recall of 0. Only minimal changes to our meta classifier, so we had one student which tried to improve it, but so he failed for various reasons, not necessarily technical ones. We improved the methods in terms of usability by requiring that all methods must use the same blast results, so that you can now predict all the gene ontologies for all the with all the methods in only one blast run. And you can actually now download MetaStudent for Debian. It is accepted in Debian actually. And also we introduced a free parameter, so which database to use for our predictions. One curious detail was that we found out that using the latest database with the most annotations as templates did not only not improve performance of our methods, but actually decrease them for the MFO ontology by 15%. And so this is something that you can reuse for all sorts of prediction problems that use the Swissprot database. So Swissprot now follows a very different policy than it did a few years ago, by really trying to correct every annotation that is in there, and really not considering, so not putting too much weight on preserving the old annotations. So you could get the impression that not much is going on there, but in fact a lot of things are going on there, in particular in terms of functional annotations of proteins. And to give you just some statistics, so this measures the difference of the GoTerm annotations between 2011 and 2013, and around 68% of all the proteins that were annotated in 2011 had lost annotations in 2013. And much of this was due to a term called protein binding, which is now completely removed from the database because, well, all the proteins bind something. But even if you do not consider protein binding, still about 40% of all proteins had actually lost annotations, so were annotated in a wrong way. Okay, but going back, yes?
But you said it's better to use the older version, right?
Yeah.
So it's better to use this wrong
it's true.
Annotations that are now deleted.
Yeah, it's a total contradiction.
Oh.
But
so it's not really good that they deleted it, right? Or Yeah.
That depends. So if you apply a more rigorous quality standard to your annotations, you might remove annotations which are dubious but may still be correct. So may still turn out to be correct in the future. So this is one way you can explain it. But still, so this is not good news, right? So generally for predictions. All right. And also we did not now only consider annotations that came from Swissprot, but also from the EBI. So those are basically two independent annotation groups, and also all groups combined. So all the annotation groups combined, and again, everything is under construction. I don't know the details of this myself, but what I think explains it the best are the following distributions. So this is the distribution of the sequence identity of all the templates to the best targets as they come from the EBI targets. So if you compare this to the first kappa, in first kappa you had sort of a hill in the middle, and then again an increase towards 100%. This is basically very different now. So there are a lot of targets from the EBI which did not have any similarity to the targets that were already annotated. But also this region here is quite overrepresented. And if you compare that to Swissprot, so Swissprot not only looks very different to a few years back, but also different to the EBI targets, namely that really in the period where annotations could accumulate for kappa 2, the experimentalists only looked at proteins that were already quite similar to the ones that were already annotated. And I will now show you 6 overall recall precision graphs. We had much more submissions than for the first kappa. So 121 methods in total. I only show the top 10. So let's look at the first ones. So the Swissprot for the MFO targets, we are at 10th place right here. Let's go further. So for the EBI targets, not in the top 10. Rather bad performance actually for BPO. What you can generally observe here is a huge improvement compared to the first kappa experiment, which might be due to the easier targets. But these regions are definitely also, in absolute terms, very usable. So this method is really impressive from Chandler, for example. As I said, for BPO, as in the first kappa experiment, performance is generally reduced, also because BPO is much more difficult to predict because it is much more terms that are possible. But for the cellular component ontology, we actually made it to the top, the first place, and you should not criticize first place, of course, but so we are at this curve which goes here, and as I explained earlier, so it is easy to get a good maximum F1 score if you perform good in the mediocre regions, and this is exactly this little hill here, which made the difference compared to all the other methods. And also for the EBI targets, we are we performed quite well at 4th place here. And if you look at, so what I've just shown here are the ranks of meta student among the top 10 methods. So sometimes we did not make it to the top 10. This is why I looked at the results again myself and looked it up where we actually stood. So at For example, as I've shown in the first place, so for the SPRO targets for the SwissPRO targets on 10th place, for BPO we are doing rather bad, and for CCO rather good. And for this allegedly improved measure that I explained, so that corrects for these issues, our rankings generally go up, which is again a very good sign. And if you would, for example, look only at the methods that made predictions for all ontologies, would look at all the targets that could be predicted, and then would calculate a rank sum and compare this to other methods, we would end up at 4th place out of 55, which is again quite a nice result. Okay, so this concludes CAFA in general. Any questions left? No.
Do you know, is there going to be another one?
I definitely think so, yes. So we are growing, so there should be one, but I don't know when. A topic that is the last topic for today, which is something I've not presented yet. Actually, I haven't seen anywhere else actually, and is also somehow generalizes the whole CAFA concept. And again, a question to you. So this is an evaluation that we did ourselves before the CAFA meeting of our 3 methods, and you can see here. So one method A in green here, up here, and the same method in the CAFA experiments is down here. And I ask you to give me reasons how such a difference in the evaluation can happen. So what is it that we did wrong here? Yes?
Yeah, it depends on what proteins you use for your evaluation, or even for the training program.
For the training, we used randomly sampled proteins from the entire SwissPRO database, and used the rest as templates.
But then you use the same, so this trained method you put into CAFA. So
yes.
You did not train differently?
No.
Yeah, then it should depend on the proteins you use for evaluation. Or did they use the same?
Well, there were also SwissPRO proteins.
Okay.
Right?
But not the same?
Not the same proteins, but so we used 10,000 proteins for our evaluation, and SwissPRO used on the order of and CAFA used, I don't know, 800 or something. But so these numbers should definitely be high enough in order to rule out any sort of statistical fluctuations because of small data sets. So it's definitely not the size of the data set that made a difference here.
But they gained it differently, the proteins. Like, how did you get to 10,000 and how did they get to 800?
We randomly picked from the SwissPRO database 10,000 proteins, and then these proteins were removed from SwissPRO, and what remained was used for blasting, right? For prediction. Essentially the same principle as in CAFA. So the only difference is that we did not sample randomly from the SwissPRO database, but we had to stick with the proteins that were newly annotated. But if the experimentalists had picked the proteins that they analyzed randomly, this again should not have made a difference.
Okay.
Yeah.
But did you first reduce the redundancy of the database?
We did not, but CAFA also didn't. As you know, I won't show this again. So if you look at the distribution of the sequence identities, so as I've just shown, you see that there is no redundancy reduction going on because quite a few targets are quite similar to the ones that are already annotated. Yeah.
Do you follow a different target function?
Following different target function.
Depends on the difference because of the different target function.
Could be, yes. This is actually a good point. Yeah. So one issue could be that the historic distribution in the SwissPro database has changed over time, so that in the past different functions were analyzed than they are analyzed now. So in the past, for example, much enzymatic activity, now more I don't know, ionic transporters or something like that. Okay, but more issues.
The experimentalists did not choose their proteins randomly, but they like
Okay. So
experiments with them. Maybe.
yeah, that's certainly true. So as I've shown already on the other slides, so this is not random. Okay, so if you would choose your proteins randomly, you would get sort of a distribution like this. So this is definitely not random. But I would argue if targets really look like this, we should actually perform better than what Kappa showed.
There is perhaps also this authorship bias, because some authors go terms are not they are subjected.
Subjected. What do you mean?
Some authors might use different GO terms for the same thing.
Oh.
So there might be a bias.
But it is so extreme.
Yeah, so the Go terms are actually assigned by annotation groups which mine literature, I believe, most and also first and foremost. So this should be actually quite consistent. But it's true. So a certain fraction of Go terms also changes over time because they're simply annotated in the wrong ways, or just some curator read it in the wrong way. But the influence should be rather minor, actually. I personally believe the biggest issue was really the evidence codes, for example, of the Go terms. So what type of experiments you would allow in Kappa, and what type of experiments you would allow for us? So in Kappa, only very reliable experiments, and in our evaluation, we basically used all the experiments that were out there. And this in this case really made the difference, I believe. And
So there were like these evidence codes only standing for like annotated by homology or something. So you used this as well.
Yeah.
So like you did not check for anything there.
So we excluded electronic annotations, but the entire rest we used. And and this can really make the difference. And so all the students, of course, they hadn't worked with the ontology, but also the exercise leaders, as us, we had no prior experience with the ontology. And to some degree, even the Kappa organizers. And this is striking because you don't cheat. You are convinced yourself that you're correct with this curve, and then this turns out to be actually false pretenses. And this is what this last topic is really about. So bias in computational biology is basically everything non-formal, every non-mathematical, non-logical thing that you can do wrong. Every lack of power in your data, anything that corrects or your accuracy estimates over time. So you develop your method, for example, in your thesis, you get really good results, and you check back 10 years later, test the method, and suddenly performance has dropped. So why is that? Also, I could call it what comes after publications. So this sort of phenomenon you have seen for decades throughout computational biology over and over again for various prediction methods, for various qualities of papers. So nature methods, science papers are affected as well. And it's generally something people really don't like to talk about, especially if they're scientists, because this puts their also their current results sort of in questions. And you wouldn't believe how many different types of biases that there are. And I will explain some of them now in a little bit more detail. So first, one that you have already learned about, that methods tend to improve if sequences are similar between training and test set. You have a bias by experimentalists to only study proteins that they somehow already, so that they are confident with. So, and these proteins are usually similar to the ones that are already annotated. The big problem, however, is that if you look at the almost 100 million proteins that we have in UniProt, most of them aren't similar to the ones that are being studied, to the ones that are already annotated. So in tests with cross-relation and new data, if you don't look out for this bias, you easily overestimate your actual performance. And so the only solution to this is really to always consider sequence similarity and the test and new data as the cover people are working on right now, I assume. Okay, another question, another sort of bias, experimental bias. So we have discussed this already, but a very different type of bias now. So assume you wanted to know how many eukaryotic proteins have more than one domain. So domains are functional parts of proteins that can function on themselves. And most of the domain definitions come from the PDB, because in the PDB you can see what is a domain and what is another domain. So how would you answer this question if you really wanted to have this question answered for nature in general, not necessarily only for the PDB? Yes.
Did you have to use a database other than the PDB, because the PDB is biased for one domain proteins, because they're experimentally more easy to determine? So
okay,
you'd have to take, I don't know, maybe a sequence database and then try to map accordingly to the PDB and use only those.
Mm-hmm. Exactly right. Yeah. So I repeat it shortly. The protein. So the PDB is a very old database, and historically, structure determination was limited to very short proteins, as a result, you have an extremely high bias towards short proteins and single domain proteins in the PDB. So if you would simply look up the distribution of domain numbers from the data that you get from the PDB, this distribution would be wrong. So what you actually need to do is you need to somehow generalize what a domain is, and scan for every domain through entire proteomes and see how much of the protein is covered by this domain. If this domain covers the entire protein, then you have a one domain protein. If it does not cover the entire protein, then you have a multi-domain protein. Is that clear? Any questions? Yeah. So again, experimental bias for historical reasons. So as I said, crystallization may have only been possible for parts of your protein. And interestingly, so I believe you are working on the Ebola virus in your exercises. If you go to the PDB molecule of the months, have you seen that? No? So there is an article about the structure of Ebola, and you find exactly this problem there. So that some parts of Ebola are structurally resolved, but some other very important functional parts are not. You will find also experimental bias in very different types of experiments. So for example, all the known interactions between proteins, they are biased by protein localization, because we can only detect interactions for proteins that are in certain localizations, which is also due to the experimental methods that we used. And also, of course, proteins need to be expressed on quite a high level in order to really be able to detect the interaction. Of course, also, as you've probably already heard, so high throughput experiments tend to have an extremely large fraction of false positives. And again, you won't read about this in literature usually. It usually comes to display if one group does an experiment and another group does the exact same experiments, but they come to totally different results. Yeah, so fixing this, all of this, unfortunately, makes you requires you to really read in depth into the methods, talk to a lot of people, and it is essentially something that you cannot really account for, and is probably one of the biggest reasons why older methods do not perform as well now as compared to new ones. Another type of bias, so-called, I call it statistical bias. It's not a good term, but yeah, I couldn't find anything better. So oftentimes you will work only with very limited amounts of data, so that, as you've already discussed earlier, then you need some sort of statistical test how large the chances that you got your results randomly. So you can use for that standard error, McNamara test, Fisher test, rank sum tests, various types of rank sum tests. So they are available for all types of predictions. However, authors of various papers do not always use them in the best way. And this is an actual comment from a very renowned scientist who I do not want to mention now by name, but so he recommended basically to use standard deviation if your results look better, if errors are large, and use the standard error if you want errors to be small. It's basically. Do you know the difference between standard deviation and standard error? Anybody?
Isn't it just like a square root or something, or divided by something?
Yeah. Okay. So
the deviation is the standard error squared.
No.
No, it's something.
Standard error is standard deviation divided by square root
of the number of samples. Yeah. So it's something very different. If you have four randomly drawn numbers, and 5, 7, 8. So you have a mean of 2, and then your standard deviation would be something on the order of 1 to 2, and it would not change if you would draw more samples from the same distribution. So if you would now go on and draw more numbers, I don't know, 5, 6, 7, 8, 9, 10, so the mean would roughly stay the same, and the standard deviation would also roughly stay the same. But the standard error then describes your trust in this mean. And your trust in the mean increases as your sample size increases. Okay, so the standard error would go to 0, in fact, if you draw more, whereas the other two numbers stay the same. So this is a totally different statistic then, and really, and it is really used. But even though this is an extreme difference, they are basically used in various ways throughout the literature. If you really don't. As they should not be used, and it's also hard to detect often. Sometimes they don't mention it at all, so they just say plus minus, and then you can guess whether it is standard error or standard deviation, or they only mention it in the supplementary materials, for example. Also, if you don't log out, for example, and draw random samples from the PDB, so PDB is a very special database in that it contains, I don't know, between 500,000 and 1 million proteins. But if you redundancy reduce it to only representative sequences, you go down to a level of 10,000 proteins. So it is extremely redundant. And if you would really only sample randomly from the PDB and use this set as your test set, you would have extremely so you would measure the same protein several times. And as I said, so these statistics, they are influenced by the number of samples that you have in your data set. But if you simply duplicate the samples, then this obviously doesn't tell you much about statistical significance anymore. And also an interesting fact about ROC curves, about the difference of ROC curves and recall precision curves, is that ROC curves always look smooth, so they can never decrease. So this is because of the way they are calculated. Whereas recall precision curves, you sort of see the sample, the underlying sample size by fluctuations in the curve. So if you have if you compare this again, so these are very smooth curves based on 10,000 samples. But here it is not so smooth anymore. So you have a lot of fluctuations here in the lower recall region, and this is actually because of the small sample size. And if you would let's say you had actually true negatives, so true negatives is the requirement for recall precision curves, which you do not have here. But if you had them, so you couldn't really see this difference, because recall precision curves really steadily increase. Solution really, knowledge of statistics, honesty, and of course peer review is supposedly help, but is supposed to help, but doesn't always help. Finally, rather machine-oriented bias, namely training bias. So if you have some sort of machine learning model, I'm sure some of you have already heard about it. So you can usually push it to a point where it doesn't make any mistake on the training error, on the training data. At such a point, overfitting has occurred, but you can also cheat by optimizing free parameters, not explicitly on the test data, but also implicitly by repeatedly checking what type of way of training works best on your test data. The ad hoc solutions to this is usually so splitting the training set in the training and so-called cross-training or parameter optimization set, and then really not touching the other sets at all. However, any type of study where you try to predict or analyze something usually has parameters associated with it, and even simple things such as blast parameter or, for example, first clustering, then predicting, or then first predicting, then clustering, and then going on can make a small but decisive difference in your evaluation. And it is also, as happened for us, of course, it is also easy to fool yourself. So this is really on the border between statistical advances and fooling yourself that you have actually made improvements. Another bias, I call it class bias. So another question. So assume you have some sort of evaluation where you get 99% of all the queries right. So when would this not actually be a good result that could be published?
If you have 100 samples and 99 of them are, or 1,000 samples and 999 of them are negative anyway, and then the last one is also classified negative, so would that still be 99%?
That's quite correct, yeah. So the answer is if the occurrence of one class is in fact greater than 99.9%. So we have 1,000 samples and 999 of them are in one class and one sample is in another class. In this case, you have, if you would randomly predict, you would already immediately get a 99% accuracy. So the only way to improve is really to predict all the ones correct. This is not an artificial scenario. So for anything related to protein interactions, for example, you are even worse than are in even worse regimes than this. And so the only fair evaluation then is really to learn and train on the natural distribution as it occurs, and not to artificially try to equal out the classes, because if you would, for example, if you would do a cross validation on the training set here, and you would get, for example, so of all the green samples here, you would get 50% right. So this means 50% of the actual green samples are actually predicted as green, and the other ones are not predicted as green. And you would then take this classifier and apply it to this test set, and you would make 50% error on the green class. This would already be worse than random performance. So you should have, if this really is your true distribution, you should always train on this distribution. Another one, circular information flow. So sounds totally silly, but it happens more often than you think. So you have one type of data A. You use it to predict or annotate data B, and then a few years pass by, and another method comes out that uses data B to predict or annotate data A. Obviously, if you do that once or a couple of times, you will improve, because information that is used to predict something is also used to, for example, evaluate something. So you go circular and circular and improve, but as soon as new data comes along, you suddenly hit rock bottom and are back in reality. And one curious example for this, is for example, if you use mass spectrometry data to annotate protein complexes, then you use the protein complexes to annotate. So then the gene ontology, for example, looks at the literature published about protein complexes and uses it to annotate proteins with CCO terms, with cellular component ontology terms. Then some bioinformatician comes along and thinks it might be a good idea to use Go terms to predict protein complexes. He, of course, has no idea that actually protein complexes were used for the annotation of the Go terms, and is happy at first that he gets so good results, but as soon as new data comes, he will snap back to reality. And even better, so an encore is if he would be using this initial mass spec data actually to validate his predicted complexes, then the circle would be perfect. All right, I believe this is the last one. Yeah. No. So-called interaction bias. This was discovered in 2012 and affects basically any sort of protein interaction predictor that has been published before that. So you now have about 50 to 100 papers published in various journals, Nature Science, which are put into question. And he used quite a neat trick, but so the author of this who made this discovery used a neat trick, but it's essentially his method is that the accuracy for predicting a new interaction depends on whether A and B, either of the two or none of the two, were used for training. And if you have a predefined interaction network and you randomly sample from this network and use this random sample as your test cases, then most of them will be in this first class, because in the network everything is typically well connected, so that if you take out one interaction, the protein still stays connected in the network, and this way again falls into class C1. The problem, however, is that most proteins in nature, as I've said multiple times, are not similar to proteins that are already annotated, even in protein interaction networks. This way fall into class C3. And this makes things really complicated. So you now have to not only look at the positive interactions, but also at the sequence similarities between them and differentiate between various classes. And in order to summarize it all, so it is very difficult to avoid all the biases that I just described. So it may be even more. So you will experience that it might be even more difficult than to develop an actual prediction method, or it may not be possible at all for experimental sorts of biases. Computational biology is definitely not the only field that is affected by all of this, and advise to you that whenever you read papers that try to predict something, don't take the numbers they give you for pure gold, but be critical about this. All right, so that's it, and I thank you very much.